Well, good morning, and thank you for joining us today. I am Laura Parent at the CTSC of Northern Ohio. I am pleased to have you join us today for our Spotlight series, our September session. Today, our topic is UG3 grants and collaboration opportunities, but of course, we've got some special guests with us today. And our host and alternative presenter today is Sue Marasco, our Assistant Director of Grant Proposal Development, excuse me. So thank you to Sue for leading the efforts today and kicking us off. And she will introduce our special guest, Dr. Jeffrey Loeb. So thank you, Sue. Off to you.
Thank you so much, Laura. I'm so excited about our guest today. Dr. Jeffrey Loeb is a champion of translational science with a strong reputation for successful multidisciplinary program development. Collaboration with both academic and private industry to produce important research and new approaches to neurological treatments, including pioneering a systems biology approach that links genes, proteins, molecules to epileptic activities in the human brain. Furthermore, he is one of the pioneers in the field of growth factor signaling in motor neurons and a developer of novel patented therapeutics for a variety of human diseases, including the development of an innovative drug targeting technology that could significantly improve the treatment of brain disorders and cancer. Through years of NCAT support, including a successful UG3 UH3 mechanism, Dr. Loeb provides insight into innovation through collaboration. Dr. Loeb currently serves as the department head of the University of Illinois Chicago Department of Neurology and Rehabilitation. He received his MD and PhD from the University of Chicago and went on to complete a residency in neurology at Massachusetts General Hospital in Boston, then followed by fellowship training in epilepsy and clinical neurophysiology at Harvard's Beth Israel Deaconess Hospital. At the same time, Dr. Loeb held a faculty appointment at Harvard Medical School, where he performed research on nervous system development in the Department of Neurobiology. His clinical and scientific careers have been seamlessly commingled. He's worked to not only be the best neurologist and epileptologist for his patients, but harness his knowledge to a strong basic and translational science commitment so that ideas, technology, and research programs stream back and forth between the clinic and the laboratory. A major goal is to learn constantly from patients and their diseases in a rigorous scientific framework that generates new hypotheses and then test them. It is my great pleasure to welcome you, Dr. Loeb, and thank you for that. I will take one more moment before turning this over to you to remind everybody that our collaborators work together at the CTSC and serve as a catalyst for high-quality clinical and translational science and transformative research to improve the health of those in Northern Ohio by collaboratively providing education, training, services, resources, programming, funding, and more to our partners, researchers, and surrounding community. Dr. Loeb, it's great to welcome you because you are an example of the best that translational science has to offer. And I'm going to stop sharing.
Well, thank you for that exorbitant and wonderful introduction. Let me, oops. Let's see if I can pull this up. So I'm going to tell you several stories today and how we've kind of used team science and different mechanisms through NCATs and other mechanisms to really understand human brain disorders and come up with new and innovative ideas on how to treat them. And a lot of this kind of evolved in my career. About 6-7 years ago, I kind of took over the informatics core at our own CTSA program as the director. And this really comes from trying to integrate many different types of data together in our various research. So I kind of came up with also an alternative title for the presentation. Artificial intelligence is only as smart as your data. And I actually give a lecture to the medical students who all go through this to say, what is AI? And I don't really spend much time teaching AI. They kind of know what it is to do ChatGPT, but what goes into it is what's really critical. And that's what we're going to talk about today. So just kind of just refresh everybody of what is AI, what is unsupervised learning. You can develop these giant computer systems with billions of data elements, and hopefully, if it runs long enough with enough power on the grid, you can tell the difference between a squirrel, a fox, and an owl. Now, this is unsupervised. You give really no information, and the computer algorithm has to kind of figure all that out. Now, if you help it out with a supervised model, and you basically structure the data, and you define here's an owl, here's a squirrel, and a fox. You can use a much smaller computer, smaller data sets, and actually get the results much more quickly. So the point is, if we can basically start with better structured data, other than the messy stuff that comes in from our clinical data warehouses, we might have a chance of doing AI a lot better. And so, can AI be harnessed to understand brain disorders and choose the right treatment? And as I mentioned, there are challenges even in diagnosing diseases. When we go through diagnosis codes, many of them are wrong. As a clinician, I put them in myself. Sometimes I have to bill before I know what's going on with the patient. The biggest data is often the messiest and inconsistent. And again, what goes in is what comes out. We don't really take advantage of the fact that we have different types of data. We have everything from medical records to imaging to electrical brain recordings, and we don't routinely integrate those together. That creates a more powerful and more unique data set for each individual patient. So the bottom line is we need good data for AI to work, and how do we go about getting it? So the approach we've taken is through something we call the University of Illinois Neurorepository. As I'll tell you about it, it's kind of a human brain bank, but it's really about all the data that's associated with that brain tissue that comes out that makes it really valuable. So our approach is to choose small, highly focused projects at one disease at a time. Get subject experts together, clinicians and scientists, to say, what are those key data elements that you really need for that particular disease? Then develop clinically useful ways to store and integrate silo data. And this is going to be important when we talk about the bottom point, which is sustainability. Once the grant ends, how do you keep people funneling this data in unless it's actually clinically useful? The clinicians are not going to spend their time to funnel that data and integrate it the way you would like it done. Then you can apply your AI. Always important at the end of this, you have to have a means to validate. So if you make a prediction, a statistical prediction, you have to have a way to go back to the tissue, to the genes, to the proteins, and validate what you found. And again, one of the key things that I always worry about is sustainability. What happens when that grant ends? You made this big database. You spent millions and millions of dollars on a database. Where does it go? Does it just throw into some big data lake, or does it stay structured and organized? And is there maybe a desire from the clinicians, from the researchers, to support keeping this moving forward? So when all of you think about doing a project that involves clinical data, you go through a series of steps, such as identifying patient cohorts to test your hypothesis, determining if the cohort has the data you need, because sometimes those patients don't have the MRI studies or the EEG studies or don't have the lab data that you would need. Then you have to get an IRB. Often you have to do the IRB first, which is a big challenge because you don't know what data is there, and then you have to go through this reiterative process. Then you download all this data to REDCap. Once you have the data, you have to need ways of visualizing or dashboards to see the data, both on individuals and groups. The data is often a mess, so cleansing procedures are really critical, particularly before doing AI, cleaning up the noise and the things, the data that's wrong, the diagnosis codes that are wrong, as we talk about. And then you do your statistical analysis or AI, validate it, and hopefully get results. So the stories I'm going to take you through today revolve around two tools that we've developed to help this process. The first tool, which I'm going to talk about later, Adjust AI, helps automate this process to get those cohorts and data that you really need very quickly and efficiently. And Intuition is the data visualization and analytical tool for a specific project. And I'm going to start talking about Intuition first. So as was mentioned in the long introduction, I am a clinical epileptologist, and really patients who don't respond to medications, we have the ability to remove brain regions surgically to cure the disease, but we have to find those areas where seizures start. To do this, we use intracranial EEG, so often either removing the skull or drilling holes and putting electrodes directly into the brain in a three-dimensional framework to know exactly where the seizures are coming from in the brain. So here's an example of a patient who has intracranial electrodes. These are grid arrays that are sitting on the surface of the brain, and you can identify the regions under each of these little discs or electrodes where the seizures start, and those tend to be the areas that need to be removed surgically to get a good outcome from this procedure. Now, obviously, you have to make sure you don't take other things that are important so the patient's still walking and talking and things like that afterwards. So it's a very, very intense procedure where you collect tons of data. Now, in addition to the seizures that are fairly infrequent, we look at the brain waves 24/7 for about 7 days or more. And you can see at each of these 1 second intervals, very frequent epileptic discharges. Again, each one of these are these different electrodes that seem to form these spikes. And they're very large areas that basically are constantly spiking between the seizures. And one of the questions we have is what is that relationship between the spikes that are occurring very frequently between seizures and the seizures themselves, which the clinicians are more worried about, because they had a convulsion or they crashed their car or something as a result of it. So we had developed tools to mark these spikes so that we can digitize these things. We take the data, we plot it back on the patient's own three-dimensional surface, again, bringing both imaging data and the electrical data together in a single frame, where you can see areas in red have a high frequency of spiking between seizures, areas of green have a low area. So for example, here's a patient that we had done surgery on and actually had a fairly large resection with a number of tissues that were present. What I've done here is marked in red circles here the areas of spiking, and the larger the circle, the more spiking there is, but also the areas where the seizures start. And so for our systems biology approach, the questions we have is what's different about those human brain areas that produce seizures that produce spiking? And we always have to have a control. And within these studies, we have an area that we also recorded from that was electrically silent, that was removed as part of the surgical procedure. We don't take any extra control tissue. If it's not there, we don't use it. So you can see here the electrical patterns of the spikes on the upper right, and the control area, and then the seizures. Then we can actually do high throughput studies such as differential gene expression, proteomics, metabolomics, to compare the control to the seizure, control to spiking, pathway analysis, validation, animal models, and again, try to drive these targets to clinical trials, which we are doing right now. To do this, every block of tissue we take, we want to have a number of questions. We want to know what's going on histologically. So we have half the tissue that's fixed for histology. We want to know what's going on molecularly. So we do transcriptomics on alternating strips, proteomics, metabolomics, etc. So to make a long story short, we've been doing this for a number of years. We compare areas of high spiking and low spiking. We map certain pathways such as MAP kinase and CRAB, localize it to different layers of the 6-layer cortex, identify neuronal changes. When we do the same thing for seizures, we get actually a very different group of genes, which was kind of surprising to us, that the areas that produce seizures and spikes have different patterns of gene expression, implicating different pathways, different layers, and different changes to ion channels. So again, this is discovery-based work by bringing many types of data together to try to find these potential both biomarkers and therapeutic targets. We can then build models of what might be going on in the brain in areas that produce spikes in these superficial layers with MAP kinase, and with seizures with deep layers that are involved in mTOR activation and produce repetitive discharges. But the point of what I was bringing this up isn't just to talk about this research, but to talk about the data and how we bring this distant data together. It's very challenging. We don't have data systems that bring together everything that you need, such as the electronic health record, the imaging, electrical activity, and then when there's tissue, ideally you want to know exactly where that tissue came from, as we've done here. So we have to register that in our three-dimensional algorithms. We then can store our histological data on the system, and then any data that comes from our omics, our genomics, proteomics, and metabolomics. And in some ways, as a physician, this is what we do all the time. We look at the records, we look at the past medical history, we look at the imaging, we look at the EEGs, and then it rattles around in our head and we go, aha, I know what's going on and what treatment I should give you. So that's how we came up with the name Intuition for the software, because it really integrates many different types of data, hopefully that we also do as clinicians, but not just data for clinical use, but also data for research, because knowing exactly where that tissue comes from on imaging, on physiology, on histology is really key. And you can imagine that even outside of the brain, if you're studying cancer, wouldn't it be ideal to actually see the same histology, genomics, and imaging for that same piece of tissue? And actually, well, the project I'm telling you about, the UG3, during the UH3 period, we're actually funded to expand this towards cancer as well. So this brings us to the Chicago Epilepsy Collaborative Project. And this basically stems from the idea that, we can only do so many cases at one given site. These are very intensive surgeries. A very busy program does 50 cases a year. That's a lot. And in big data, we need more data. Plus, when you start having heterogeneity in patients who have, different genetic backgrounds and things, you may need more patients. So the idea was, can we basically take this intuition platform and create a federated system and put this at multiple sites, and not just have the software, but also train people and getting that structured data and processing the tissue exactly the same way. So you could bring tissue samples together from multiple sites. They were fixed the same way, they were processed the same way, so you can actually do an experiment and not worry about this variability. So the title of the UG3 grant is Integration and Interoperability of Complex Data and Tissues from the Human Brain. So let me tell you a little bit about the UG3 UH3 mechanism and what we've done. So this is just an outline of the data structure that we have at each of our sites. We pull in electronic health record data from clinical notes, medications, complete neuropsychological data sets, imaging, and EEG. We then bring these together and co-register them, like in the pictures I showed you, the 3D maps showing where things are. And then we keep track of tissue that comes out as well as the outcome of the patients. And all of this lives in a secure clinical environment. Through de-identification, we can actually pull data through this and then have a constant flow of de-identified data for research. So we built this at the University of Illinois in Chicago, and now we're building it at Rush University, Northwestern University, Lurie Children's Hospital, and University of Chicago. And Chicago is a big place, and we have 3 CTSA programs. So this is a collaboration of 3 CTSA programs and 5 sites to bring this together. To do this, we need a team. So the team-based approach is really good involvement of the clinicians. The clinicians are the ones who have to spend that extra time, whether it's in the operating room or in the EEG suite, to pull that data together. So we have site PIs who are epileptologists at each site. We also have folks like Biswajit Maharathi, who's our chief architect, who actually, you can see, is on the call today. Visualization design experts, tissue processing. Again, we need to train all these sites to process the tissue in the exact same way. We have our follow-on project with brain tumors. We have our consultants and neurosurgeons, neurologists scattered here, as well as additional experts and federated platforms, Brett Below-Jones at University of Chicago, in medical informatics, Alan Wu at Lurie Children's, genetics, genomics, and again, brain tumors, plus ontology consultants. So bringing all these folks together, it's like herding cats, but it can be done. And we actually have been quite successful at getting people engaged, which is a challenge. So we have regular meeting structures to do this. Our entire group meets monthly and yearly in person. And again, Zooms, I think, has made it easier to do these things. The data elements, we spent a lot of time more at the beginning defining what are those data elements for epilepsy patients. What do we need for the database? What data elements on EEG? What data elements on MRIs? And how do we pull these analytical 3-dimensional models together? Weekly informatics, IT, visualization, user interface, privacy. IT and informatics is huge in this project, and as I'll tell you, there's a lot of challenges in doing this, particularly among multiple sites. And again, the goal is to also disseminate this as well. Weekly meetings for research staff to learn about data upload and tissue banking. So again, those who are collecting tissues need to get trained. Fazia, who does this, actually physically goes out to the other sites when necessary to do some of their surgical cases as well. But even things like co-registering and knowing exactly where the tissue comes from, it's not trivial, and it's a fair amount of work. So building tools that makes it easier helps do this, but it does take extra work. So these are the aims, specific aims of both the UG3 portion, which is the first 3 years, and the UH3, 4 or 5 years. So build and deploy a federated intuition platform at multiple sites with standardized data. Basically perform multimodal data and tissue harmonization using existing and new standardized protocols, training programs for data and tissue collection, making user-friendly analytical tools for brain imaging and EEG. So if these tools actually help our epileptologists and surgeons take care of their patients, people will use them. And if they use these tools clinically, we have that data available to us for research. And then it's pristine data because it has to be. We cannot make mistakes with the patients, as opposed to pulling it retrospectively and guessing what's right and what's wrong from charts like we usually do. So once we have the federated platform built, we have to develop a use case, and we're going to focus on temporal lobe epilepsy patients to create a project across all 5 sites and measure the spike propagation and seizure networks related to lesions. Another thing for dissemination and sustainability in the UH3, convert our in-person training programs to online training programs, collect tissue from patients with brain tissues so we can show that this platform will work for other diseases in addition to epilepsy, and then find other CTSA hubs that want to employ this and use this, as well as solicit private sector support or interest from this, because I think you can see that the data and the tissue can be extremely valuable for the pharma industry as well. Because once the grant ends, how do we sustain this? That's the big question, and all of the things we do. So here's some examples of how we localize the tissue. This is the patient with the recording electrodes. We actually use these arterial trees that you can see here as like fingerprints to make sure that the tissue matches. And here's a tissue temporal lobe block that came out. You can see it's fairly faint here, where the electrode locations were 60, 59, etc. And then the tissue is subdivided. It's broken to pieces. Each block of tissue, according to a specific electrode, is further cut in half, where half of this tissue is frozen and the other half is fixed to do histology. We do the histology first to see the actual architecture of the tissue, and then use that to design a further dissection of the frozen tissue for our RNA, protein, and other studies. So in addition to standardizing our tissue processing methods, we developed, actually, Biswajit, who's on the call, the architecture for how this whole system works. And again, this is a big challenge because each site has its own rules and security issues, particularly when dealing with PHI data. Each site has access to their own PHI data, but of course the federated system doesn't see anything except de-identified data. So for a given site, you have your basic architecture for both storing data, querying data, but you also have a central routing app through the federated platform that can reach out to each of the sites and pull a subset of de-identified data that can be queried by anyone at any of the sites. So again, as I said, there's a number of challenges when doing such a project, and we have to sort of think about these questions. What types of data can be shared and can't be shared? Can tissue be shared? Who owns the data? Who owns the tissue? How do we prioritize the use of data and tissue during the grant period and then after the grant period as well? How do we, share data when, after it's published? How do we publish? Can we work with commercial entities, pharma companies, or not? Who owns the patents or commercialization interests? What happens to the data and tissue after the grant ends? To do this, we've set up a governance board, which basically consists of all of our site PIs and the key players in this project. And we developed a number of legal agreements between all the universities, which took longer than we thought it would take. Surprise. So we're very pleased that the material transfer agreement to be able to take our software and put it at 4 other sites has been done, and we're now in the process of disseminating the software so people can start loading data. But then once we have that, how do we share? How do we share data? How do we share tissue? How do we use this to work with our governance board? We're working on this agreement right now. One of the key elements that I instituted in this to make sure everybody would play along is that any site has an opt-out option. So if you have a project and you want to use your tissue for something, nobody from this governance board can come and say you've got to give up your priceless human brain tissue for our projects or our collective communal projects. So it's really who else would participate in this? Same with data. If their R01, if their grants are dependent on certain data sets and you give it away to someone else at some other institution, so you can always opt out. And I think that encourages people to participate. And for things they're not working on, certainly I think majority of things people are willing to share. So these are some of the challenges that we're working on. Again, privacy issues have also been a challenge. We have to track our progress. This is a cooperative grant with NCATS. So we have quarterly meetings with our program officer, Tom Radman, our PPRs. So we have to create our list of milestones for all the sub-aims and check boxes of how we're doing or where we're falling behind and how we're going to catch up on those things. So I'm not going to do a demo of the Intuition Epilepsy today, but I'm going to do a demo of another project that is further along in terms of the visualizations, and it's a little more fun. So this has evolved a project that was funded by the Department of Defense to try to understand not treatments for epilepsy, how do you prevent epilepsy? What happens when you injure your brain and make some people more likely to develop epilepsy than others? So this is a DoD-sponsored grant with Cure Epilepsy, which is a patient foundation that helps fund this. We got 6 centers together and multi-investigator teams to study diverse aspects of post-traumatic epilepsy. So this is our UIC team. Again, we have epidemiology, we have our clinicians, coordinators, imaging, informatics, and we actually have an animal model in this as well that we parallel this to. And the subarachnoid hemorrhage is something you can study as a component of traumatic brain injury because just about every TBI has subarachnoid blood. So instead of studying the injuries themselves, we're studying subarachnoid hemorrhage, which is often caused by aneurysms, and they develop epilepsy at about the same rate, 10 to 20%. So the patients who get subarachnoid hemorrhage, they end up in the intensive care unit with all these beeping machines that are constantly spewing out data in real time, just tons and tons of data in the whole course of the ICU experience. In addition to data that kind of pops into Epic and as tabular, we have EEG data, like we talked about before, that evolves over time and changes. We also have CAT scans that are done almost every day on these patients, showing the blood as these white areas in the brain and how it progresses and changes over time. So all these data elements, these multiple data elements, we pool together to build a longitudinal multimodal data set and use that multimodal data set to try to predict who gets epilepsy and who doesn't. And the goal in the future is to say, can we use this data almost in real time in the future to help manage patients and prevent them from getting epilepsy or other complications? So we took our Intuition platform for epilepsy and translated it into the Intuition Neuro ICU platform. So here's where the demo comes in, and hopefully it works. So this is the home page that pops up. We have our patient data, our animal data, all in this database. So we can go back and forth between animal models and human. But if we just pull up a single patient here, we can get some basic demographic data. And the timeline overview gives a nice view of all the data that we pull in automatically from the data warehouse via Epic. And there it goes. So basically, we talked about diagnosis codes. So the admission diagnosis code, as you hover here, is non-traumatic subarachnoid hemorrhage. But look at all these discharge diagnosis codes. They're all right here, and they're all listed here. Patients during this 3-week hospital course have a lot of different procedures done. You notice the patient was admitted on January 1st of 2000. We basically took all of our patients and basically lined them up so they bled in their brain. It was a very bad Y2K. We had a lot of subarachnoid hemorrhages on that day. So we can see what happens over time and compare between patients. So these are procedures, lots of clinical notes. We have the raw data from those as well. Lots of CAT scans, lots of blood tests. We bleed these patients a lot in the ICU, a lot of vital signs and medications. We give a lot of different medications. This is the total list of medications from a single patient. So it's really hard to grasp what's going on. This is just a nice overview, but if you want to see what's going on a day-by-day basis, you can pull up a dashboard and see what's happening in this patient and choose what you want to look at. And one of the things we're quite interested in is looking at the spinal fluid, the CSF, and particularly at the red blood cells. So we can see what the red cells are doing, and you can see they're coming down nicely after the bleeding in the brain. We can pull up what's going on with the protein in the spinal fluid, because that often parallels what happens to the red cells, but you can see it's on a different scale. So to see it, we create visualization tools to normalize it. So you can see these two come down very nicely together. We were a little surprised when we looked at the inflammation in the brain and look at the CSF white blood cells that there was this delayed peak. And actually, this turns out to be one of the variables that predicts who gets epilepsy after subarachnoid hemorrhage. Say you want to know what's going on in kidney function, you could pull up the creatinine, whatever you want, you can see here. But this may be important in how we're managing the patient. So what are we doing with the blood pressure? So here's all the systolic blood pressures on the same time scale. Here's heart rate, etc. We can bring all these things down. In the neuro ICU, we actually look at the intracranial pressures. We have catheters in their brains that actually allow us to read the intracranial pressure. And you can see here, very shortly after admission, there was a big spike. That's not good. We also record something called Glasgow Coma Score, which is a neurological exam assessment of how sick the patient is. So we can put that side by side. And if you zoom in on this area here, you can see when the intracranial pressure goes up, patient gets sicker. When it comes back down, patient gets better. So this could be actually useful to a clinician who's actually looking at this. We also want to know, are we doing the right or the wrong things with all these medicines? I showed you all these medicines. Let's just look at all the medicines. And you can see we have a visualization tool that lines them all up. You can see every dose, every medication that was given and when it was given. So this allows you to go back and forth with your medications, what's going on in terms of vital signs and lab tests in real time. And these are all basically tabular data, so numbers that you can pull from medical records and just upload, instantly, even in real time. More complicated are things like notes. So if you go look at the clinical notes, you can see this patient had 468 notes. It's a lot of data. You could, though, screen and say, well, let's look at the EEG reports, and you can see there's 16 EEG reports. And then we can pull those down. We also have the ability to actually take the raw EEG data there and pull those up as well. Radiology, again, every day at 3 a.m., patient goes down and gets a CAT scan of the head. One of the major challenges clinically is how do you compare the first CAT scan to the third, to the fourth, to the 15th? How do you make those comparisons? So to do that, another talented MD-PH engineering student built some cool tools to visualize the brain over time as movies. So the white areas you can see here is the blood that should not be in there in the brain. On day 2, you can see a new bleed that came back here, this new blood that happened down here. This is just taking a slice and creating a movie through a co-registration process. This bleeding goes down, and then you can start seeing the swelling of the brain come down, because those black areas are the fluid-filled spaces, the spinal fluid areas, the ventricles that are now starting to fill with spinal fluid again. There's air that appears because they took the ventriculostomy out. And if you're impatient like I am, you can grab this little bar and go back and forth. And eventually this will line up directly with all the other data, so you can see what's going on in the brain, what's happening on the EEGs, what's happening in the different aspects of it. So that's Intuition Neuro ICU. The study we did, we recruited 162 patients. We ended up with 134. Of those, 15 developed seizures, chronic seizures or epilepsy. We then developed really sophisticated ways that are beyond my abilities to look at every one of 58 variables we looked at to see if there's a difference between the red line, which are the patients who developed epilepsy, and the black line of patients who didn't develop epilepsy, to see if there's a statistically significant difference. So you can kind of see, if we zoom in on some of these, that those were sicker with a worse Glasgow Coma Score. The red line is below the black line. Hemoglobin is lower. Eosinophil, and total surprise is really, and again, the fact that we did it longitudinally, we never would have picked this up if we looked at admission eosinophils. It's really at the time of discharge that the eosinophils went up. And again, these are variables that are consistent with patients developing epilepsy. Prothrombin time was elevated. Red cells, not surprisingly. And then another surprise, this delay inflammation that we see here of the white blood cells in the brain. So we can take these individual variables, put them together in a logistic regression model, and improve our current predictions, which are the black line to the blue line, in terms of by putting multiple variables such as the Glasgow Coma Score, inflammatory indexes, eosinophils, and total blood in the brain. If you add the white cells in the spinal fluid, you improve the predictive model even better. So this will be important if we ever want to recruit patients for a clinical trial to prevent epilepsy. We want to choose those patients that have the highest risk by doing this type of work. Another fun thing we did, independent of developing epilepsy, we just did a correlation analysis at early time points, at middle time points, and delayed time points in the hospital. So any two variables that have a positive correlation would show up as a darker blue. If there's a negative correlation or inverse correlation, it would be a darker red. And you can see if we zoom in this early part right here, that the more blood that you have in the brain, generally inversely significantly correlates with a poor clinical exam. So that kind of makes sense, and that's a nice control. But then you see things like the intracranial pressure here, and if you zoom down, you can see some things that correlate strongly with increased intracranial pressure, and that's CSF, white cells, and neutrophils. Again, something you wouldn't expect, but something, wouldn't this be great if this were running in real time and a little red flag popped up and say, do you know that this patient's getting sicker and these variables correlate with that? Could you do something about it? So always thinking, can we take this research project and turn it back into something clinically useful? And this is actually one day we had a fire alarm in the building, and I was running out. Of course, I had to take my laptop because I was working on this stuff, and I had this program open, and one of the neurosurgeons said, "What are you working on?" And I showed them this and said, "When can I get this?" So this is, again, how do we get this type of software back into the clinic so that it improves clinical care, reduces physician burnout? Simpler notes and dashboards would be wonderful to practice medicine, seeing this every day as opposed to this lengthy garbage that we see all the time in our Epic notes or Cerner. No offense to either one. And then once we have data structured this way for clinical use, it automatically feeds for our research program. So we don't have to sit there and mine it and curate it and clean up the mess, because it's going to be pristine from the get-go. So I've talked so far about what we do once we have the data. I didn't talk about how we get the data. Many of you probably go through the process of identifying these cohorts and testing hypotheses and getting IRBs and then changing the IRB because the data and the patients that you wanted didn't exist, or it was the wrong data. So then you got to keep going back and forth, and maybe a few months later, you can actually get your data. And we were struggling with this for quite a while. And when I was the director of the informatics core at our CTSA program, in the previous cycle, we succeeded in getting an optional module for Address AI to basically develop software that helps us pull this data in an automatic way, even without an IRB to start with. So I'm going to tell you a little bit about Address AI right now. So again, we need data that feeds into our intuition projects, Address AI. So here's the next demo. And it was having a little problems this morning, so hopefully it's still working. Yes. So this shows that I've done a number of searches already. We could either do you could tell me some favorite things and do some searches, or I could show you some existing searches. So let me just show you one first, and if we have time, you guys can play, because it's really a lot of fun. So I had done one to look at patients who have both stroke and migraine, but then also have seizures. So trying to identify a patient cohort for a study I was interested in. So this is the 3 panels you get when you bring this up. Let me see, here it is. Okay. And the first thing you do, you can see the 2 panels here. The third one's over here. I'll bring that up in a second. That you identify the patients you want. So you come in here and you filter your demographics. And we had asked for patients age 18 to 99. So you can select the age range. You can say, do you want sex or race or ethnicities? Do you want to do it on certain dates from the medical records, 10 years ago, 20 years ago, however, 30 years ago? I think we have about 20 or 30 years of data in our data warehouse, and that all comes up and pops in here. Once you've done that, then you identify your cohorts. So you switch here to the terminology, and the first search we did here, we did a search for migraine. So if we just type migraine here, again, without knowing any diagnosis codes, you can pull up everything in the database, from SNOMED codes to ICD-9, ICD-10, anything with migraine. Now you can either select all of these, or you can pick individual ones. I've selected them before, and then you basically drag these and bring them over to your cohort graph. I can go back and I can look at this and I can see, oh, well, I excluded retinal migraine. I didn't want to include that in this group because that's the eye and not the brain. But I included all these others, and you can change it and move that around. And you can go back and forth. Then I wanted to add another cohort. I wanted to add stroke. So then I came here and I did a search for stroke. And it pulls up again all the SNOMED, IC codes, and I can select and then I drag this over and I choose and because I want patients who have both stroke and migraine. And I did the same down here for seizure. I wanted to see those who had all three, winners, because there's probably some relationships between them. So again, we have the and see. Well, you could say or initially, so you pool certain groups together and then add and later. Once you do that, then you click execute, and that basically goes to the data warehouse and pulls down all of the patients that you've identified in this cohort builder at this point. So now I'm going to lose this field here and focus on this one. So it's a lot to ask to have seizures, migraines, and stroke. So we only got 105 patients, 22 male, 83 female. We have the racial ethnicity. You can also see this if you want in a graphical format to see what you have here, or a tabular format. You can also have a timeline where you can basically see when were those patients seen in our hospital and healthcare system. And the beauty of this system, it's all de-identified data. You do not need an IRB to do any of this work. You just need an ID from the university. And this is taking a little bit longer. Let me see if it has its issues again, like this morning. It's got to go through a lot of records to do this. I see Biswajit's on the call. He told me everything would be working. All right. The other thing that's nice about this program, changing subjects, is that it generates a SQL code. So everything, when your informatician goes to the data warehouse and pulls the data, they have to sit there and manually write these codes to basically pull the data with inclusion, exclusion criteria, diagnosis codes. This automatically generates a SQL code. You can give it to your informatician, they can pull it down. So let's say you've gotten all this data. You can then search a number of things like, do they have other diagnoses? Do these patients have CAT scans, MRIs? Do they have certain procedures that were done on them? What medications do they take? You can see all that. You can graph it. I mean, we can try to see if that's going to work now, like the medications that these patients took. And again, it's just these 105 patients. Normally, you can see all of these things, but it's been giving us the proverbial rotating donut. CBiz, this is what I was telling you about. We're in the process of moving the platform from here. Oh, we got something. But that's not the drugs or medications. Let's see if it works now. Maybe while it's working on this, I can show you the next steps. So once you identified your cohort, you know what data you want to get your data somewhere, right? So we next go, you save your cohort, and then you request your data. And this brings you to a new screen where it allows you to walk through, summarizing your data, your project, what data fields you want to collect, where do you want it delivered, who has access to this, a summary statement, and then you'll see it'll actually generate a data request authorization form. So here's just the summary of what we've done. You go to the next, it gives you the project details in terms of the cohorts, and do you want de-identified data, identified data, do you want data or just cohort identification? It then says what data elements you want, and you can actually choose. We've chosen before all these different measurements from blood tests, from procedures, etc., of the data that we want to get. You next have to come here and say, where do you want the data delivered? Do you want it in REDCap? Do you want it in a secure compute environment, Box Health, other? Then you add users. You can say, I want to add certain people to this, informatician, collaborators, co-PIs. And then finally, it'll give you a summary of everything, sort of like the SQL code. And you can basically generate a PDF for a data request authorization form that basically puts this on. You can hand this to the IRB, and once it's approved, hopefully a week later, if it's de-identified data or it's exempted, you push a button and it delivers the data to your REDCap or wherever you want it to go. So let me go back here. So key about Address AI is you do not need an IRB. Anybody with a UIC identification card can use it. You require a login to a VPN and a login to Address AI, so you have to set up the security. And because we have this, we have to train the next generation. And this is an amazing tool to sit there and let our medical students play with it. They don't need an IRB and can ask questions while they're on service. They see patients with certain conditions and they want to know some certain things about it. They can get all this information. And if they decide they're interested, hopefully they'll push the button and generate a data request authorization. They'll get the data and do more research. So we want to encourage more people without strong informatics backgrounds to be able to ask questions and do research. So integrating this into our medical curriculum is something that we've done. So Intuition continues to expand. In addition to epilepsy surgery, neuro ICU, we have a rare disease module that we're building out, and also one on neuropsychology. A lot of those kind of can integrate with each other over time. And again, in addition to Address AI feeding data, we're working for the neuro ICU to do automatic data feeds through things like HL7 and FHIR to feed data automatically into the system for clinical use, so that we then can just dump it from the clinical situation into the research situation in a de-identified way. Again, I want to highlight Biswajit Maharathi. He's the guy who really does all the work and built all these software platforms and architected them. And Fazia Amir, who directs our Neuro repository. And there's a lot of other people. Again, I mentioned the UG3, but if I had to do a slide with all of our collaborators and so forth, that would go on forever. But I wanted to end and have time, which I think we do, for some discussion. What are some take-home messages or discussion points? I hope you agree that medical data is a mess, and you can't do AI without accurate clinical data. Software with standardized data elements that's clinically helpful and reduces physician burnout could help solve this problem and actually get us data that is actually standardized and useful, that doesn't require so much cleanup. Team science across institution is challenging. One of the things that's really helped is really engaging the clinicians, that they see this as valuable, both for their clinical practice and their research programs, and getting them engaged and coming to meetings and herding the cats is really critical. And dissemination is, again, part of the plan. So in this talk, and hopefully other talks I will give, and others will give, we want to encourage people to reach out to us for Address AI, Intuition platforms, and collaborate and work on these federated platforms as well. We're eager to share this with other folks. But also we're interested in sustaining it. So once the grant ends, who pays for, some of these things? Who pays for the data? It's really important that we think about how we keep this clinical utility so that people find it valuable. Maybe hospitals will pay for it because it helps physicians do their jobs better and more efficiently. And then at the same time, it gets data flowing for our research. So we're actually working towards licensing and commercialization of the software to a company so that we can sustain it and support it. It's a lot easier to keep it going that way than it is to say, well, let's apply for another UG3 grant or another CTSA, get the optional module, which don't exist anymore. So you've got to apply for something else and then you're down for a while. So working with industry could be a potential advantage. So I'm going to stop here. I'll leave these questions up and see if anybody has any questions. Thanks.
Oh, thank you so much for this presentation. This was unbelievably helpful and checks all the boxes for dissemination and implementation science, along with all the other great things. I did want to make sure we got two questions asked of you. Priya, unless you want to ask it yourself, I will ask about the sleep data. Okay. Priya was wondering, is there sleep data included? And how do you include it if there is? And if not, why not?
Of course, sleep is really important, particularly in brainwaves. Things change dramatically when you're sleeping, so we pull, all of the EEG data, which includes awake and sleep. So it's quite simple to go through and find segments of sleep and find segments of wakefulness, um, and correlating that with what's going on. I think in the ICU with all of the drips and things like that, patients don't show normal sleep patterns at all, and that can be really detrimental to outcome measures. So the continuous data in the ICU, which I think you may be referring to, and is important for the epilepsy surgery patients, we have the continuous monitoring also that has sleep data in as well.
Fantastic. And the other question for Madeline was, what methods of data validation are being used to ensure accuracy and consistency of the model?
I mean, we have to go back and take a look, obviously, and do some validations to make sure the data is accurate. And we do find inconsistencies in any of our data projects that we do. Even with standardization, there's still a data cleanup process that you have to go through and check some things. And sometimes things just don't make sense, and you got to put red flags on them. For example, in our rare disease project, we asked people, and we have 8 different sites, fill in the past medical history. Okay. And this is a project for Sturge-Weber syndrome, where a lot of the patients have seizures and headaches. And whoever was filling out the forms, it said the past medical history was like 30 or 40% had seizures of our past medical history, and of our family history as well. And that just didn't make any sense. So I think people were mistakenly filling out the forms wrong. And I think getting it from the chart will help if we can pull it directly from the chart, as opposed to asking clinical researchers to go through the chart and interpret and put things in the right place. So that's one, trying from an upfront way to increase accuracy by pulling data directly from clinical records, by structuring the epic notes in a way that they're standard data elements in all the epic notes. So the clinicians have to fill them in to get their notes done. And then we just pull them right from those epic notes, and we don't have to have somebody translate or copy and paste and have a chance to make mistakes. But in either case, you have to have a cleansing and a cleanup and a validation round.
Well, fantastic. Those were the 2 questions in the chat. I've got one from Mary Delansky, but I want to make sure that anybody else on the call that has questions has a chance to ask them. So please unmute and ask your question if you have one.
Don't be shy. I know Zoom, it's hard to do, but did it all make sense to everybody? It's clear? All right.
Yeah, it was.
Oh,
Please go ahead.
hi, Dr. Loeb. First, thank you for this wonderful, really insightful presentation. Kudos to you for all of your work on this. It's been a really wonderful lecture to listen in on. Following up on the data validation question, is the methods of validation mainly in your entry points of data, or are you using any other form of validation when you're, trying to procure the results at the end of using this model?
I think the answer is both. I mean, the more that we can do up front, the easier it is downstream.
Oh,
But
yes.
yeah, we clearly have, and in terms of method, I don't know if you're speaking of like a specific methodology or something. Usually there's so much data. When we start asking questions of the data and we start seeing inconsistencies or things that don't make sense, then we go back, and then we have to go back and look at the raw data and see, was there a translation problem? Was it written down incorrectly? Was the diagnosis code wrong, etc.
Yeah, that's mainly what I was asking is what you're describing sounds more like data proofing, where you're making sure the original entry data was accurate. And then more looking at if there was any, my background's in statistics, so I'm more on the statistics side of things. So seeing if there's any type of, outlier detection, methods you were using, regression analysis, just more of a curiosity,
Yeah,
or
no, with each
everything's
project, I think I
automated
showed you, some of
validation?
those longitudinal curves. We worked with one of the professors of statistics at our institution because it was way above my pay grade in statistics, which is pretty low. And we show the spaghetti plots, we show the averages, we show, we calculate it, multiple different ways to convince ourselves something is really true. But yeah, I mean, this is really, again, hopefully when we get the data, the raw data directly from, the machines that are generating it, we don't have to worry about the transcriptional errors in terms of the data. But there's going to be a huge variance anytime you work with human data, and there are going to be outliers. But that doesn't mean you can't learn from it.
Makes sense. Thank you, Dr. Loeb.
Are there any things here that might be of interest in collaborating? How do you do your informatics? How do you extract your data requests? Is it done manually by informaticians? Do you use I2B2? Do there's another platform out of University of Washington some people are using. Nobody hear from there? For those who are aficionados, all of our data from both, we had Cerner and Epic, was fused together during my tenure there into an OMOP data model, which is a very standardized model that all of us use, as many of the national programs use. So we basically converted our entire data warehouse and funneled it into the OMOP data model. But I mean, the address AI would work on I2B, work on any data model. It would take some time to kind of set it up and match the fields. But it's all set up for OMOP.
In the comments we have UH uses, which is the University Hospitals, uses Trinetics and Epic Slicer Dicer primarily for de-identified data.
Yeah, has anybody tried to use Slicer Dicer? It's not easy. It does, it doesn't have the capabilities. So I mean, that's why we needed to do this. You don't have all the depth of data. We have the entire data warehouse, which has even the raw notes. You can actually do that, LLM models and probe the notes as well.
The additional comment is it's not a great system, unsmiley face.
Yeah.
So
Well,
thank
I
you
should
for
say
this.
we're eager to share. So, I'm happy to talk to your folks and work together.
Which is why we wanted to have you on. Thank you so much for all of this. In the last 2 minutes, Mary Delansky, who runs our workforce development module, wanted just a few comments about what team science has meant to you. And we talked about herding cats, but maybe some of the other collaborative.
Yeah. No, I used to, when I started my career, I mean, as I said, I think we talked earlier, I had no fear. I could do anything myself, and I was going to try to do everything myself. And that just doesn't work. I mean, just even, statistical, analysis. And so the challenge is getting people together, getting them excited and enthusiastic about what you're doing. Each folks as part of the team needs to feel they have a personal stake. They have something that they're gaining from that interaction, other than, oh, I get some grant money. It's got to reflect on their personal interests and excitement about what they're doing. And you need to be aware of those things and engage people in such a way that they feel really part of the team, that they have a say in what's going on. There's some team science that's just run by a leader and everybody just follows. It's a much more fun when everybody is arguing and you divide up the tasks with experts in their own areas, and they each have an important contribution. So I think that helps. But you have to be excited about the project.
Well, we couldn't have ended on a better note. Thank you so much, Dr. Loeb. This has been fantastic. I appreciate you so much, and look forward to connecting you with more people.
All right. Nice to meet you all. Bye.